Hi, my name is Havu, and I'm a grad student in Professor Jason Owen's lab at UCLA. I'm very thankful to be able to present today at RSGDREAM, and my talk is titled A Framework for Group-Wise Summarizations and Comparisons of Chromatin State Annotation. Epigenomic signals such as different histone modification marks, DNA hypersensitive size, etc., can be a really powerful source of information for annotating the genome because they can reveal a lot about the locations and dynamics of regulatory elements. You get my point. So here I'm showing a snapshot of the genome where each of these tracks is showing the signal of different chromatin marks. And given that we can profile different chromatin marks across the genome, methods such as chromatin has been developed to learn the combinatorials and spatial patterns of these chromatin marks and then classify regions of the genome into different chromatin state. This is the chromatin state annotation, and I'm highlighting two windows that are annotated as the red state corresponding to TSS and promoter activities because they share the same kind of patterns of signals of chromatin marks. And these chromatin state can be characterized and given really biologically meaningful names through model parameters and enrichment with external genome annotation. So the motivations of this project came from the fact that there are an increasingly large compendiums of chromatin state maps covering large biological space, such as those generated by APMAP or BroadMap projects. And then it is desirable to have a summary chromatin state annotations and probabilistic estimate of the state assignments for a group of related samples. So for example, right here, each of the rows is showing the chromatin state annotation for different samples, or what we also call reference epigenome. You can see that samples from the same kind of cells or tissue groups are grouped together. So it will be nice to just have one summary chromatin state annotation for each group. And we note that an obvious approach to this problem is through counting the state frequencies across a sample, which we call the base count method. However, this approach does not leverage the information of state co-occurrences across the genome. Um, Let me give you another example. So here I'm showing the chromatin state annotation for 7 brain sample and 5 ESC sample. An example output would look like this, where we can get the summary chromatin state annotation for each of these group of samples. And we also get the summary state assignment probabilities, where each of these tracks is showing the probability of each state being assigned across the genome. And a potential extension of this is to score the differential chromatin state assignment between two groups of multiple samples. So an example would look like this, where each track is showing the differential chromatin score. Negative score as observed in this track means that the brain samples are more likely to be in these states compared to ESC and vice versa. So with that, we introduce our method CS-REP. This is an example input data where each rows correspond to each genomic positions. Each columns correspond to different samples. In the cells, the color of the cells is showing the chromatin state. Um, so at its core, CS-REP is an example of multivariate logistic regression classifier, where each classifier is really trained to predict the chromatin state in a sample using the input data of chromatin state annotation in the remaining samples. For example, this top classifier right here is trying to predict the chromatin state annotation in sample 4 using data from sample 1, 2, and 3. The output of each classifier is the matrix that shows the probability of each state being assigned each positions in the target sample. So we can just average these output samples to get a final output of CS-REP, which shows the probability of state assignment at each position for any samples in the group. For example, a new sample associated with the same cell or tissue types as the input sample. So to calculate the differential score between two groups of sample, here I'm showing the example input for two groups, group 1 and 2. We pass the data through CS-REP to obtain the summary state assignment probability for each of these groups. We can then just do a simple matrix subtractions to obtain the differential chromatin score, where positive scores um, in blue mean that uh, the group 1 samples are more likely to be in this state in these positions compared to group 2 and vice versa. Using CS-REP, we generated data of summary chromatin state map for 11 groups of sample from RoadMap. and 75 group from APMap, and this data is available for download or for being viewed on UCSC Genome Browser. So you can look at your favorite loci here. Next, I'm showing some evaluations of our method. Here, I'm using data of chromatin state annotations for 64 samples from 11 tissues group from roadmap. And for each of these groups, we conducted leaf 1-hour cross-validation analysis, where we use a summary state assignment probabilities to predict genomic locations that are associated with individual state in a held out sample. And we obtain the AUROC as our matrix of evaluations. We compare CS-REP against base count. In this plot right here, I'm showing the AUROC in recovering location that associate with the state number 1, which is active transcription start by state. Each data points correspond to each of the 64 sample, and you can see that CS-REP shows higher distributions of AUROC compared to base count. And this pattern was actually consistently observed across all the 18 states in our model. Next, I'm showing an analysis regarding gene expression. So here we collected data of chromatin state annotations and gene expressions in 8 cell type from roadmap and 64 group of cell type from APMap. And for each of these groups, we calculate the Spearman correlations between the summary assignment probability for the active transcription start size state, annotated transcription start size, and the average gene expressions of the corresponding gene. You can see that whether you use data from roadmap or APMap, the Spearman correlation resulted from CS-REP tends to be higher, so implying that the summary state maps from CS-REP shows better correspondence with gene expression. Next, I'm showing example output of differential chromatin score at the MT3 genes. This is input chromatin state annotations for 7 brain sample and 12 blood sample. And we pass these through CS-REP to generate the differential chromatin score. And you can see, for example, this top track right here is the differential score with respect to state 1 active transcription stat side. It's all negative, meaning brains are more likely to be in this brain are more likely to be annotated as an active transcription stat site in this region compared to blood. And we know that the AMT3 genes has been shown to have distinctly higher expressions in brain cell type according to GTEx data, so which is consistent with what we observed with CS-REP. Next, we show another evaluations where we use the differential chromatin scores between ESCs and brain samples to predict the genomic positions that overlap with either ESC-specific or brain-specific peaks of DNAs, H3K9AC or H3K27AC. And we compare the performance of CS-REP against 4 other methods. 3 of them are existing published method, and 1 of them is the base count method, where we just take the difference of the sample frequency between the 2 groups. And we note that compared to existing approaches, CS-REP offers several advantages. First is that it's calculate genome-wide calculations of differential chromatin score, so it's not being restricted to just an individual locus. Second, the method generates differential score at the resolutions of input annotation data, which is the highest resolution possible. And we generate individual score track for individual state, which increase the interpretability of the model. So here I'm showing the matrix of evaluation is AUROC, and we're showing the AUROC for different methods in recovering regions that are associated with brain-specific DNase peaks. And we know that because DNase H3K9AC and H3K27ACs are all chromatin marks associated with active genomic regions. So we specifically look at the performance related to promoter or enhancer states. And the top 3 AURC are highlighted in bold and underscore. And you can see that CS-REP tend to result in higher AURC. And this pattern was actually consistently observed across our context of comparisons. So in summary, we introduced CS-REP, which is to probabilistically summarize the chromatin state maps for a group of samples and score the differential chromatin state activity between 2 groups of samples. The approach is an example multivariate logistic regression classifier. We show that CS-REP summary chromatin state map can help predict locations of states in held up samples and shows correspondence with group-wide average gene expression. We also show that CSF differential score can help predict group-specific chromatin mark signal picks, and we generate data that for different tissues group from roadmap and APMap, and they can be downloaded or reviewed on UCS Genome Browser. Our method is on GitHub. It's implemented in Python and SnakeMake, and we hope you can check it out and give us more feedback. And this is a link to our manuscript that just been accepted to bioinformatics. With that, I'd like to thank all my co-authors, my advisor for his guidance, and my lab member for useful discussions, and my funding resources. I'm happy to take any questions.
Great talk. So I did not quite understand why you had that regression, and if you were only going to average the outputs ultimately, why not just average in the data itself?
Oh, yes. So averaging the data itself is the method that we show by simply using the frequencies of state counting, which is the base count method that we presented.
Okay. Oh, okay. That's the
Yeah.
base, and you show that it performs better. So
Yeah.
in that step, if there is a particular predictor in a particular state that is poorly predicted, do you deal with that as some sort of uncertainty in the score? You have a predictor there, and sometimes for some of the class tasks it will work well. For the other tasks, no, in your approach, go where you show the logistic regression to predict the fourth sample from the other three, right? At that point, if you found that your method does not, it's not your method, the predictor, the MLR doesn't work for a particular task, do you do anything about that?
I guess no. We
Okay.
kind of put it in a pipeline. We
Okay.
didn't really have a step to check that part.
Okay.
So no. Yes,
All right.
but it will be
Yeah.
nice to check that part. Yes, that's
Okay.
a really
All right.
nice
Thank you.
input. Thank you.
I actually had a question about this slide, so it's great you flipped to it. The question is specifically about the input with the one-hot encoding. Am I correct in thinking that you're not in any way incorporating the fact that some chromatin states are more similar to each other than others? And if so, have you thought about maybe revising the way you do the input to incorporating that and seeing if that helps?
Yes. So yes, the input data is one hot encodings of state assignment in remaining samples, and we try to. So the reason why we use this MLR is so that by training an MLR, we learning that certain states um, are more similar to each other because across the sample they tend to co-occur at the same genomic location. So we hope that the MLR is actually doing that learning.
I see. So the model does it internally, hopefully.
Yes.
Cool. Thank you.
Yeah. Thank you.
One quick question. So you are comparing these groups of samples. Do you actually jointly compare, or is the matching of the states actually done as a post-processing step?
Could you repeat the questions, please?
So you have groups of samples. On each sample, you're running chroma getting states. But how do you compare the states across different groups of samples? Is that like part of the an interpretation of the states? Like this is an enhancer, this is a promoter, this is blah? Or is there some joint training going on?
Yeah, so that's a good question. Yes. So because our data is really consortia data, so they've been uniformly processed, meaning the chromatin state has been like jointly learned across all the samples. So that means that the model, the chromatin state definition is shared across all the samples.
Okay.
So that's how it's useful in this case.
Okay.
Yeah. Thank you.